Well, good afternoon, everyone. Welcome to today's seminar. I'm Andre Drent. I'm professor of plant pathology at Centre for Horticultural Science in Kwafi at the University of Queensland. And I will facilitate today's seminar. Move to the next slide. I'd like to acknowledge the country. So the University of Queensland acknowledges the traditional owners in their custodianship of the lands on which we meet, and we pay our respect to their ancestors and their descendants who continue cultural and spiritual connections to country, and we recognize their valuable contribution to Australian and global society. There is a few housekeeping rules. So the presentation will last for about 40-45 minutes, and then there is time for questions. Please put your questions in the Q&A and not in the chat functions. I will read out the questions at the end and combine questions if necessary. You move to the next slide. Now, today's seminar is by Jane Ray about blood disease. Now, Jane called me up in about 2017 and was extremely persistent to undertake a PhD in our group. She already had more than a decade of experience as a plant pathologist in the Northern Territory. So she started in 2018. Now, what actually is a PhD? I think it's a couple of four things. It's like a unique learning experience, the freedom to investigate something new. It's probably an opportunity for some exciting travel. And it's a demonstration that you have what it takes to be a researcher and also publish your findings. Now, Jane chose this blood disease problem in Indonesia, which involved a lot of travel to Indonesia and a long stay there. And she managed to hit all those four goals I just mentioned above. It also ended up with a very good thesis and a handful of publications. So Jane, please, the floor is yours.
Thanks, Andre. Thanks for that introduction, Andre. Today I'll be presenting blood disease, an emerging threat to banana production. So after I've finished this talk today, I'm very happy to discuss any details and answer any of your questions. So as Andre mentioned, this talk is based on my recently completed PhD thesis at the University of Queensland. And I'd like to acknowledge that this work was conducted as part of a team of people. And I'm super grateful for their input and support throughout this journey. I'd like to acknowledge the contribution of my supervisory team, in particular the input of Professor Andre Drent and Professor Siti Subantia, who not only made this project possible, but they also made it a success. I'd also like to thank our funding sponsors and our partners. Now, we all love bananas, and they're the world's most consumed fruit. Now, Indonesia alone produces around 7 million tonnes of bananas a year. Bananas are an important staple in this part of the world, as about 50% of Indonesia's population are smallhold farmers, many of whom live below the poverty line. Now, the Kepok banana variety is popular in this part of the world, and it's highly susceptible to banana blood disease. Now, blood disease is reported from Indonesia and Malaysia, and it causes substantial crop losses. So what is blood disease? Blood disease is a bacterial wilt that affects bananas. It's caused by a bacterium, Ralstonia syzygiae, subspecies celebensis. The disease causes characteristic symptoms. The fruit pulp rots, there's red-brown vascular discoloration, the leaves wilt and become necrotic. It's a really savage-looking disease. Now, local transmission of blood disease is believed to be predominantly through insects mechanically transferring the bacteria on their bodies from diseased to healthy male bells. Tools, soil and water have also been implicated in disease spread, whilst long-distance spread is hypothesised to be through the movement of contaminated planting materials. Now, these are all assumptions, and they're mostly based on field observations. So on people going out in the field and going, oh, I think that's what's happening with banana blood disease. I think that's what we're looking at. And it's also based, so these assumptions have been also based on the biology of a couple of other bacterial wilts that affect banana. And I'm talking about Moco and Xanthomonas wilt. Now, both of these have had a little bit more work done on their biology. But Moco disease, Xanthomonas wilt, and blood disease are completely different species that have all evolved in completely different parts of the world. So what I'm getting at is that we really don't know that much about blood disease. And that leads me on to my research. So the first part of my research, here we look at the geographic expansion of banana blood disease in Southeast Asia. Now for this, we first developed specific research questions. So what is the historic and current disease distribution? Are there specific patterns in the way the disease disperses? Which banana varieties are susceptible in the field? And what is the identity of the isolates causing the symptoms? And can we prove that these isolates are pathogenic on banana? Can we prove causality? Now, to answer these research questions, we first conducted an extensive literature review. So we looked at the historic literature in Dutch, in Indonesian, and in English. Then we did surveys across Indonesia to determine the presence of blood disease. Now, during these surveys, we collected diseased plants. Normally, I do isolation from the banana fruit because this is the easiest. We'd get the bacterium into culture, and we always used media amended with tetrazolium chloride or TZC media because the bacterium is distinctive on this media. So once we got the isolates, we did some basic biochemical testing. We then did molecular testing to prove their identity. Now, we also tested a small set of isolates. We did Cox postulates with them, and that was to prove that the isolates we were looking at were actually capable of causing the disease. And all of this work was actually, all of the work with live isolates was done over in Indonesia. What did we actually find? So this slide is a combination of the literature review and the survey results. It's the history and distribution and the pattern of spread of blood disease. Now, blood disease was first observed on a small island just south of Sulawesi, just over 100 years ago. So on this island, the Dutch had kapok plantations that they'd been growing for about 30 years, and these are highly productive. The disease came in and it just wiped out the production on these islands south of Sulawesi. The extent of the damage was such that these plantations were actually abandoned. Now, the disease was then found endemic in southern Sulawesi. And at this point in time, the disease was not known from anywhere else in Indonesia, nor anywhere else in the world. So in their wisdom, they put a quarantine order in place, preventing the movement of plant material from this part of the world. Now, this quarantine order was incredibly successful, and for over 60 years, there were no further detections of blood disease. The disease was almost forgotten until in 1987, it showed up in West Java. And from here, the disease spread rapidly across Indonesia's vast archipelago and has recently shown up in Peninsular Malaysia and Sumba. Now, from From Peninsular Malaysia, there are no barriers to prevent the disease from spreading across land borders and into other Asian countries. Now, when the disease enters a new area or a new crop area, crop losses approach 100%. Blood disease is certainly on the move, and it's certainly an emerging threat to Southeast Asian banana production. Now, I'd like to show you this case study on the island of Sumba to demonstrate dispersal at the local level. Now, BDB was first noticed on the island of Sumba in 2011, and the disease swept across the island in only 4 years, and it's now endemic. So the disease destroyed the once thriving Kepok banana industry in 4 years flat. Now, the other thing I'd like to point out here is you can see from the pattern of spread that it radiated rapidly from a single infection source. Now, this is somewhat suggestive of dispersal by contaminated insects, tools, water, soil, and perhaps plant materials. Now, during the surveys, we confirmed BDB in a total of 154 banana plants. We found it most commonly on the highly susceptible cooking banana pissang Kepok up here. But we also collected blood disease from a wide variety of bananas across 4 species: Musa paradisica, Musa acuminata, Musa balbisiana, and Musa textilis. The Cavendish is also susceptible. Now, at this point, there is no known resistance within the Musa. Hence, looking for ways to manage the disease is incredibly important. In our study of the distribution of blood disease, we've determined its current distribution, its pattern of distribution, the pathogenicity and the identity of the isolates, and that it has a really wide host range in the Musa. The disease has recently expanded its geographic range and is certainly an emerging threat to banana cultivation in the region. Now, if you'd like more information on this section, this work has already been published in Plant Disease. So for this chapter, we really wanted to understand inflorescence infection biology. That is, where and how infection by blood disease occurs. Now, Ivan Budenhagen theorised that infection predominantly occurs through open xylem at the male bell flower cushion. So we're talking about this part here. But this theory, it's quite a good theory, but it was not supported by any data. And that leads us on to the specific research questions for this chapter. So firstly, we wanted to establish if there was a relationship between inoculum concentration and disease development. Some basic biology. Does infection occur through the male and the female parts of the inflorescence? Where exactly are the inflorescence infection cords? Now, are the xylem vessels open, and does infection occur via open xylem vessels? Now, blood disease is exotic to Australia. So to answer our research questions, we set up a field trial in Indonesia. Now, we planted about 160 Kepok and 160 Cavendish plants. Then when they were flowering, I moved over to Indonesia for about 9 months to complete experiments on full-grown and potted bananas. The inflorescence was also bagged at emergence in silk cloth bags to prevent any unplanned infection. And this worked really well. Now, we first started with the basics. I first wanted to know if the concentration of inoculum affects the development of disease. Now, I inoculated potted Kepok plants with the bacterium diluted from 10 to the 10 down to 10 to the 2 colony-forming units per mil. This was a serial dilution. And then I injected 500 microliters of this into the stem. we found that all dilutions from 10 to the 10 down to 10 to the 2 caused disease development in all of the replicates after 18 days. Symptom development was a little bit delayed, concentrations below or at or below 10 to the 4 colony forming units per mil, but it was only delayed by a couple of days. Now, this means that only one or a few bacterial cells are required to enter the plant to initiate full infection. Now, in every case, the expression of symptoms was identical. So this is an uninfected plant, and then it slowly wilts no matter what the concentration of inoculum that was injected into them. Now, in this experiment, we wanted to know which parts of the banana inflorescence are susceptible to infection by blood disease. So I inoculated freshly revealed plant parts of female flowers, malleable bract scars, malleable flower cushions, snapped rashes, and the deflowered fingers. And then I assessed these over time, and I looked for symptom development. We isolated the bacteria, and I did PCRs on them to prove that they were caused by blood disease. Now, we showed that all plant parts act as infection cause, all of these plant parts that I had inoculated. Really important here is that we found that both the male and the female parts are susceptible to infection by blood disease. And this is important because it has implications for how we manage the disease. Now, in this experiment, we wanted to determine if the xylem are open and does infection occur through open xylem. So we conducted a series of experiments using blue agricultural dye to test this open xylem theory. Now, dye was applied externally to freshly revealed plant parts, and after 24 hours, I cut the plant down, and I found that the dye was inside the plant and staining the xylem tissue. The dye was just drawn inside the plant, providing evidence not only for open xylem at these plant parts, but for evidence that there is a really strong negative hydrostatic gradient happening. If you look here, so this is the female bract scar. After 24 hours, the dye was sucked or taken inside the plant, almost 2 meters. This was just a phenomenal result. I had no idea that the flow of xylem was so extreme inside a banana plant. Now, the plant parts with open xylem, shown here, were also shown to be infection causes in the previous experiment. So if you think about it, an insect can just visit these plant parts with a bit of bacteria on the outside of them, and the bacteria just gets drawn inside the xylem where it can start an infection. Now, Ralstonias are really happy in flowing xylem fluid. This is their really happy place. Now, all of this strongly supports the theory that infection occurs through open xylem. Now, we've shown that infection occurs through the fruit, can occur through the pedicel, but can it actually occur through open xylem at the flower end of the fruit? So we applied dye to various parts of the female inflorescence. Dye was applied to the female flowers, stigma, tepal tip, deflowered fingers. And if you look here, dye applied to the female tepal tip, and this was after 24 hours. It was just drawn inside the xylem of the plant. So dye was drawn inside the plant from all of the plant parts with the exception of the stigma here. The dye certainly wasn't drawn. This is just a bit of diffusion of the dye in the stigma. So yes, infection can certainly occur through open xylem of the flowers and fruit. We first showed that a really low concentration of bacteria, perhaps only a few cells, is enough to cause disease. We've shown that infection occurs through the male and the female parts of the inflorescence. Our data strongly supports the theory that infection occurs through open xylem. Now, with this, we can conclude that if we protect the inflorescence from insect visitation, this would reduce insect-mediated disease transmission. And what this means is that bagging the inflorescence and emergence in a sealed cloth bag and removing the male bell by snapping it off would assist to reduce transmission in the field. Now, if you'd like more details on this section of work, it's already published in Phytopathology. So for this section of work, we explore transmission of blood disease in banana. Now, we first need to determine which plant parts are actually infectious. That is, which plant parts produce sap and ooze that are capable of causing infections. Then we want to understand where the infection courts are and how transmission is actually occurring. So can infection occur through the cut surfaces generated during routine pruning and harvesting practices? The tools themselves actually transmit the bacteria. And can the disease pass from the roots of a diseased plant to the roots of a healthy plant? Does the disease pass from the mother plant through to the sucker? So to understand plant transmission, we first need to understand where this inoculum is coming from and what plant parts are infectious. So for this experiment, I collected sap and ooze from the male bell. That's the ooze there that I collected from the male bell. I collected it from the rachis, the bunch peduncle, the pseudostem, and the fruit. So that's the rachis when I'd snapped off the male bell. I collected the sap and then I injected it into potted indicator plants. And what we found is that the sap and ooze from all of the symptomatic plant parts that I looked at were all infective. Now, does infection actually occur directly through fresh cut surfaces? And these are the cuts to the plant parts, and they resemble those made during routine pruning and harvesting activities in the field. So for this, we spray inoculated the fresh cut petiole, the pseudostem, bunch peduncle, the corm, and the rachis when cut to remove the male bell. And we monitored the plants for symptom development over time, and I examined them for vascular staining once external symptoms were visible. So after 22 weeks, we found that all of these plant parts act as infection courts. So infection can occur through all cut surfaces made to a banana plant, or certainly the ones that we showed here. Now, we wanted to know if the cultivation practices of pruning and harvesting can actually transmit the disease. Specifically, do tools transmit the bacteria? Now, for this experiment, I stem inoculated plants, which I let develop symptoms, and I used the sap as the inoculum. Now, disease developed in every single Kapok and Cavendish plant that I inoculated using a knife. The bacterium was highly tool transmissible. The level of the transmissibility of the bacterium here really blew me away. I wasn't expecting it to be for us to get results like this. Now, in this next experiment, we wanted to know if root-to-root transmission occurs. So to answer this question, I grew 2 plants per pot, and I inoculated one of the plants. Now, transmission occurred in 1 pot of the 24 pots I inoculated. This is the singular plant that I got transmission in. Now, so that shows that root-to-root transmission can occur, although it appears to be infrequent for blood disease. Now, I found this result really surprising. It was not what I was expecting at all. So when this experiment was done with Ralstonia and paired tomato plants and with Moco on bananas, transmission rates were close to 100% were observed when they did this experiment. And that got me thinking, why did this not work with, what's the difference between these bacteria and blood disease? And you know, this difference may be due to blood disease lacking flagella, which is important for pathogenicity in soil. Now, we use field-grown plants here to assess if blood disease can pass from a mother plant to a sucker. So I inoculated the inflorescence of mother plants and I made sure that they were that they got infected with blood disease, and then I monitored the suckers for disease over time. Now, no disease was observed in any of the suckers after 5 weeks, but all Kepok and Cavendish plants had at least one sucker with symptoms after 22 weeks. Now, this shows that blood disease moves from the parent plant to the sucker. Now, this is important because most bananas don't produce seed and they're propagated through suckers or tissue culture. People commonly use banana suckers for replanting in Indonesia, and they move them right across the country because there's actually no restriction on moving banana material at the moment. Now, this data supports the conclusion that long-distance dispersal is through the movement of contaminated planting material. We found that saponews from symptomatic plants was highly infectious. Cut surfaces act as infection cords, and the disease is highly tool transmissible. Our data as a whole supports the hypothesis that local dispersal of blood disease is predominantly through the mechanical transmission or contact transmission of the bacteria on the bodies of insects, birds, bats, and through human activities from diseased to healthy banana plants, whilst long-distance dispersal is through the movement of infected planting material. We conclude that to prevent transmission, with this data, we conclude that to prevent transmission, we need to protect the inflorescence through bagging. We need to debell by snapping to remove that infection cord. We need to sanitize our tools, and we need to use clean planting materials. Now, this section's already been published in Plant Disease if you'd like more details. Now we move on to the final part of my talk, the diagnostics of blood disease. Now, diagnostic methods are an important component of disease management and eradication strategies. To address the current lack of validated diagnostic assays, we specifically sought to test published assays with vigorous validation parameters, test and validate a novel real-time PCR assay. Now, for this body of work, I'd really like to acknowledge the significant contribution made by Dr. Vivian Rinken Flores, who has recently published this section of work in Plant Disease. Now, the bacterium has a bipartite genome structure, and it's comprised of a chromosome and a megaplasmid. Now, there are two previously published sets of standard PCR primers to diagnose blood disease, and these target different parts of the genome. Now, neither of these, this is the BDB-2400 primers and the BDB-121 primers. Now, neither of these primer sets were validated to a particularly high standard when they were initially published, and both sets were screened on less than 25 isolates that originated from a very limited geographic range. Now, Vivian has also designed these real-time primers and a prime-time probe, and these act as a high throughput option. Now, the primers and probe target part of the chromosome. For a diagnostic test to be good, it needs to be validated. But we also need to know what we are actually validating, which is why positive controls are just so important in this process. So here we use the reference isolates originating from the recent taxonomic revision by Safney and isolates confirmed by Koch's postulates to cause blood disease. So that was done in the first chapter. Now, we sought to validate the primers to NATA standards. We test the specificity against BDB isolates from a wide geographic range and test that the assay does not amplify other strains within the Ralstonia and banana endophytes. We assess the limit of detection, and this is done through a dilution series. We look at the assay's repeatability. The assay is repeated multiple times by the same person. We look at reproducibility, and in this case, reproducibility is assessed by having a different person conduct the PCRs in the laboratory. For ruggedness, we changed the chemistry, and here we change the tag. So analytical specificity was tested with 174 isolates of blood disease from a wide geographic area, right across the Indonesian archipelago and up into Malaysia. Now, the BDB 2400 assay could only detect 136 of these isolates. That is, it gave 38 false negatives. Now, these false negatives actually came from a collection that we did in Sulawesi. Now, the fact that this assay, I mean, it clearly fails this specificity test, but the fact that this published assay was not able to detect all of the strains just shows why validating primers against lots of isolates is really, really important. So the other primers, that the 121 assay and the real-time PCR assay, could detect all strains of blood disease. They didn't amplify any of the other Ralstonias, and they didn't amplify any of the bacterial endophytes. They also did not amplify any of the samples from banana tissue, and these primers could amplify the target from banana tissue. So here we check the standard PCR assays for sensitivity and repeatability. So both of the standard PCR assays were highly sensitive. This is the dilution series here, and they were highly repeatable. It gave the same results every time. The novel real-time PCR was also highly sensitive and highly repeatable. So one of the standard PCRs and the novel real-time assay passed the validation process. Woohoo! Now these target different parts of the genome, which increase confidence in pathogen detection and identification. So we now have accurate, robust, and sensitive diagnostics available to definitively identify banana blood disease. Now, this section is published in Plant Disease if you'd like more details about the primers or the process. So through this 4-part study, we now have a greater understanding of the biology and epidemiology of blood disease. This improves the disease management options to reduce grower losses and reduce disease spread. Blood disease is certainly an emerging threat to cultivation of bananas in Southeast Asia, Australia, and beyond. Now, efforts to manage the disease and prevent further spread to disease-free areas are urgently required. And there's still so much we don't know, and so much research work that can be done to understand this devastating disease. Thank you very much for listening. I just wanted to say it was certainly a great experience working in Indonesia, and I just wanted to show you a couple of shots. So the field trial site was several hours south of Gajah Mada University, where I was based. So I made this sterile cabinet here, and this was just made from a big plastic box and perspex. And I used this at my, what we called the field laboratory, which was also actually my hotel room. And in it, I would do, I'd make inoculum for the field trial. So I had fresh inoculum. I'd do serial dilutions, and I'd conduct isolations. And the homemade sterile cabinet worked really, really well. And so this is my banana farmer, Paxidero, and on this day he's very, very happy because he's taking a haul of some bananas that we didn't need from the trial. He's taking them off to the market to sell them. Anyway, thank you very much.
Okay, Jane, thanks very much for the excellent presentation. If you have any questions, please put them in the Q&A section below, and then I will have a look at those questions. And so there is one question already from Andrew Gearing. Jane, has the bacteria moved from cultivated to wild populations of bananas? And if so, would this be a threat to wild bananas in Indonesia?
I actually believe that the evidence so far seems to show that the bacterium probably co-evolved with wild bananas in Indonesia, because Indonesia is the center of origin for, or one of the centers of origin for bananas. So I think that it probably co-evolved with the wild bananas, and then when it came into contact with bananas that were put into cultivation, that's when it became a really, really big problem. And I think that it becomes a really big problem because wild bananas are, they're quite spaced out in the forest that they grow in. And so the movement between, or the transmission from plant to plant, I would imagine would be through birds and bats and perhaps insects flying fairly long distances to move the bacteria around. But once you get this disease into a commercial situation where you've got bananas side by side, you've got, once infection's in there, you've got so much inoculum, and you've got so many plants. People use knives and they transfer the disease from a diseased plant to a healthy plant. And I think this is why the disease has become such a huge problem in Indonesia.
And there's a follow-up question from John. Is there any possibility of seed transmission between wild seeded bananas?
We haven't actually looked at that. So we haven't investigated that. But I think it's probably unlikely. Once a plant becomes infected, a wild plant becomes infected, as far as what we saw during the surveys, it would eventually lead to plant death. So, yeah, I think that transmission through true seed is probably fairly unlikely.
So Jane, I have a follow-up question, which you didn't mention. What is the risk to Australia? So is Cavendish susceptible, and what evidence do you have for that? And how much of an emerging problem could this be?
Yes, Cavendish is certainly susceptible. Both of the surveys, we found that it was naturally infected in the environment, but we also put Cavendish in all of the field trials. It is highly susceptible to blood disease, and it causes the same set of symptoms. Now, the risk to Australia, blood disease is certainly moving closer to Australia. If blood disease gets into Australia, I think it will be quite difficult to manage, because at the moment I believe that like in Queensland, the bunches are quite damaged by up to 20% of bunches are damaged by bats that come up under the bunches and damage the bananas. Now, if a banana has blood disease and a bat comes along and feeds on those bananas and then moves to another healthy banana plant during the night some distance away, it'll take the bacteria with it. So I do believe it's a huge risk to Australian agriculture at the moment, or banana cultivation.
So there's another follow-up question. Are there any other host plants who are susceptible, which are close to plantations?
We don't. So some pot trials some artificial inoculations were done by Baharuddin during his thesis. So he artificially inoculated several plants like some heliconia and some other plants. So I can't bring them to mind right now. And he reported that he was able to generate symptoms in those plants. But we haven't actually looked we haven't seen and we haven't looked at alternative hosts in the field as such. But I think that it's possible, but the risk is fairly minimal. So it's possible that there are other strains like in the Moluccas. We haven't done surveys up in the Moluccas yet, and we don't know what we would see if we did surveys up there. It's possible that the bacteria originated from there, and there are other strains that with other biological properties in the Moluccas.
Okay, well, thanks very much. Well, thanks for your seminar, Jane. If there's no further questions, can you please move to the next slide? Okay, now, thanks for attending the seminar today. Next week, we have a seminar by Professor Michael Udvardi about functional genomics of symbolic nitrogen fixation in legumes. So if you can sign up for these seminars, if you've not already done so, you can do so at the website, kwaku.edu.science/seminars. And also, if you like to have some comments or you have some feedback, you can scan the QR code on the next slide, and that will help us to improve the seminars enormously. Now, thanks very much for attending today, and goodbye. Thank you.